Now, Dr. Schram, let's talk about one of the most pressing questions clinicians have about MCEDs, the real-world experience, as there is currently only one test commercially available as an LDT here in the United States.
I think one of these posters actually was your work, and so I'd like you to start off this discussion.
Thanks for sending that right back at me. So, early real-world experience with a multi-cancer early detection test. This poster was presented in ASCO in Chicago of this year, and it looked at the first 53,000 MCED tests that were delivered by physicians currently within the United States and compared the outcomes to the Pathfinder results, which is a previous study that was done by Gallery. So the average cancer signal detected rate for Gallery was 0.95%. You could see the difference with 1.1% in males and 0.9% in females. And these were also comparable to the modeled MCED testing performance rates and cancer incidence from SEER registry. From that 53,000 individuals screened, 72 individuals had a cancer diagnosis. What was exciting to see was the percentage that were found in earlier stages. So 33% of those 72 were in stages 1 and 2, and 61% in stages 1, 2, and 3. The other part to the study was taking a look at the cancer signal of origin and the prediction, and how accurate was this test in the real world at predicting the cancer signal of origin. And from this real-world evidence, we found a 91% accuracy rate, whereas in the Pathfinder study, the prediction rate was only 84%. And from the clinical validation study, so prior to Pathfinder, called Circulating Cell-Free Genome Atlas study, it was around 89%. We did see that the cancer signal detection rate increased with age, with those significantly higher, greater than age 50. We also saw in males and females that 67% and 61% of cancer signal of origins represented cancers without recommended population screenings, respectively. So what we're seeing is that real performance of Gallery is consistent with the previous large-scale clinical studies, including the cancer signal detection rate and accuracy of the prediction to help guide us as physicians with our workup. Next, I'm going to continue and talk a little bit more about two different case scenarios that were discussed, or case studies that are of great interest. So the first was about a HPV-mediated oropharyngeal squamous cell carcinoma diagnosed using the multi-cancer early detection blood test. So this test came back positive for head and neck on this individual, an asymptomatic 74-year-old Caucasian male. This individual had a history in the past of chronic lymphocytic leukemia, and this was 17 years prior, as well as papillary thyroid cancer 8 years prior, with no evidence of disease at that time. In regards to his other cancer screenings, this individual had an elevated PSA level and was a non-smoker. Blood work and imaging then followed this cancer signal of origin that was found. A final diagnosis of HPV-mediated squamous cell carcinoma of the oropharynx. The patient underwent surgery with staging and did not need chemotherapy or radiation because this was caught at such an early stage. The next one was actually a case study of a misdiagnosis. So this study showed to us that despite high average cancer signal of origin prediction accuracy, the biological similarity among HPV-related cancers for the anogenital tract and the head and neck tract may lead to cancer signal of origin misclassification. So that's the take-home from this. Cancer signal of origin misclassification may be considered in patients with an HPV-mediated history. The use of this MCED test informed aggressive cancer workup and led to the early detection and treatment with curative intent.
Those are both really instructive cases, I think, about the power and the possible pitfalls of using MCEDs at this point in time.
So Dr. Schram, I'm going to add one more thing here, because as a physician myself, as I am talking to my patients in clinic, when I look at tests like this, the most impactful thing for me speaking to my individual patient is actually that positive predicted value. In our real world poster, I'm going to go back to that for a second, We weren't able to calculate positive predictive value yet because there were over 300 individuals that were still going through their diagnostic evaluation, and we didn't have the data of, yep, a cancer was found, or no, we're considering this as a false positive right now. So, when we're looking at a test from a whole population perspective, that's where the sensitivity and specificity are really important. But boots on the ground, us physicians interacting with our patients on a daily basis, what do I tell my patient when they do come back with a cancer signal of origin, right? It's that positive predictive value of how likely are we to find a cancer for you at this stage. So I'm excited as time progresses and for us to continue to gather the information of those additional 300 individuals that did have a cancer signal reported to them from that real world data, to actually follow them over time, for another year and see what we actually, the results that we get from the continued diagnostic workup.
Yeah, and I think it really underscores the importance of informed consent prior to any test, not just testing like MCEDs or in your case, Gallery, but in any test, what does a positive test mean? What does a negative test mean? So that we can manage their expectations and reassure them, whatever the result.
So there was another reported real-world study presented at ASCO using the Themis MCED platform, and this is work done out of China. Can you tell us some more about the report?
I'd be happy to. The Themis platform presented some interesting numbers with an N size of a little over 1,000 adults. This was a study that was done in China in patients who were considered high risk over the age of 65. None of these patients had known cancer at the time, but they were all considered high risk for cancers that are more endemic in China, specifically esophageal cancer, colorectal cancer, hepatocellular or liver cancer, lung cancer, and gastric cancer. And the risk factors for these are H. pylori, blood in the stool, hepatitis B virus, or history of smoking. This model, the Themis model, was able to determine that 58 of these individuals were found to have cancer that we might not ordinarily have been able to identify. That's about 4%. It's certainly a very interesting study, and it's a platform that deserves further investigation.
Thank you for that thorough review, Dr. Schram. I wonder when we will see Themis making its way into commercial use. With Gallery and Themis real-world results, we are looking forward to what they can bring to our patients in the future.